Fighting Blindness Canada's View Point is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by AGTC, Allergan and Abbvie Company, MeiraGTx, Novartis and Roche. We'd also like to thank Accessible Media Inc., our national media partner. Everyone here should download the AMI app, which is a fabulous resource. And you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoy this webinar and share your thoughts with us in the comment section below.
I'm really excited for our next panel, which we have three wonderful researchers who are going to be talking about inherited retinal diseases and some of the research that is ongoing, both in their labs as well as more broadly. So we've used the term inherited retinal disease before, so if people who don't know, that is an eye condition that is caused by a specific gene mutation and specifically impacts the retina. And that includes diseases like Leber congenital amaurosis and retinitis pigmentosa, and also X-linked retinoschisis, as well as many others. So in this webinar, we're going to hear, panel, we're going to hear from Dr. Orson Moritz, Dr. Bob Chow, and Dr. Gregory-Evans. And so we'll just be a quick little panel discussion. We'll ask each researcher a few questions and leave time at the end for your questions as well. So I will start the program off with Dr. Bob Chow. So Dr. Chow is a professor at the University of Victoria in the Department of Biology. His research is focused on understanding mechanisms that underlie neural and retinal development, and this includes genes involved in eye disease. And Dr. Chow is also currently funded by FBC for a project involving Stargardt disease. So we're going to focus a little bit more on Stargardt disease, but hopefully have time to delve into some of his other research interests. So welcome, Dr. Chow.
Oh, thank you very much. It's a pleasure to be here. Thank you for the invitation. And it's just wonderful to hear all the speakers presenting today. It's a really important symposium to have.
Thank you. And Dr. Chow, I think might be, has come the furthest as a speaker for FBC for this session. So a special thank you to Dr. Chow. So starting off with, if we could set the stage for some individuals who might not know, your FBC-funded research and some of your research is focused on an eye disease called Stargardt disease. So could you tell us a little bit more about Stargardt disease and how that impacts vision?
So Stargardt disease is a pretty rare retinal disease. It affects about 1 in 8,000 to 1 to 10,000 Canadians. It's the primary cause for macular degeneration in children. And as a macular degeneration, it affects primarily the central part of the vision. So as you know, the central part of your vision is what you use when you're looking at someone's face, or if you're reading a book, or if you're trying to focus on something. So an individual with Stargardt disease won't be able to have a clear image of the person's face that they're speaking with. And that can vary tremendously. It could be kind of a subtle defect in being able to see the person, or it could actually be very extreme and you can completely block out the person's face. On the other hand, the peripheral part of the vision is normally left intact in Stargardt disease. However, over time, they can also be affected.
Called Stargardt disease macular degeneration. Earlier in the day, we heard from some experts who were talking about age-related macular degeneration. Mm-hmm. Is there a link between the two types of diseases, or are they completely separate? Yeah,
so there are some similarities in that they both affect the macular part of the retina, and they are both progressive diseases. The differences are that the onset for macular degeneration comes much later. And as was mentioned earlier, it's a very complex disease. Although there is some genetic component to it, we really don't understand that very well, and there are other components that can affect the onset of macular degeneration. On the other hand, Stargardt's disease is referred to as a monogenic disease, in which we know that the disease is caused by mutations in a single gene.
That's actually a perfect segue to the next question about what are the genes that cause Stargardt disease? Is there only one gene, or are there multiple genes that cause the disease?
Yeah, so 4 genes have been identified that can cause Stargardt's. They're all kind of different in terms of their gene structure, but they're all present in the retina in photoreceptors. And we believe that they all kind of affect the same kind of pathways because they lead to the very same type of phenotype and pathology in individuals with Stargardt disease. However, the most common form of the disease found in about 95% of Stargardt's patients is caused by mutations in a single gene called ABCA4.
And in that gene ABCA4, I think there are multiple types of mutations that can occur in ABCA4. And depending on the type of mutation, does it have different impact on vision and vision loss?
Yeah, absolutely. So you can have several types of mutations that will disrupt the gene. Some of these mutations may be very, very subtle in how they impact the gene and the disease function, and others might lead to a severe effect. So in Stargardt disease, subtle changes in protein sequence have been correlated with more subtle effects of the disease, while removing the gene completely by mutation will cause a more severe phenotype. That's known.
Okay.
Yeah.
And I think the answer is probably maybe no, but are there currently any treatments for Stargardt's disease or anything that can help reduce vision loss progression?
Yeah, so there's currently no cure for Stargardt's disease. There are different approaches that can be used to slow the progression of the disease and maybe to help whatever residual vision the person has. So for instance, it's well known that one of the risk factors for progression of the disease is smoking, so avoiding secondhand smoke or smoking is necessary. There's been a correlation of exposure to blue light with the progression of Stargardt disease, and that's known from basic research that has shown that blue light will activate the formation of toxic molecules in the retina, which is the cause of the pathology of the disease. As I mentioned before, because the peripheral part of the retina is normally intact, there are other therapeutic approaches, including eye devices that can help kind of maximize the vision in the eye when the central part of the vision is lost.
So if blue light might impact Stargardt's progression, is there a way to block blue light specifically?
Yes. So potentially the use of eyeglasses with filters blocking UV blue wavelengths of light can be used. Yeah.
So maybe moving on now to some of the research that you do, Dr. Chow. I was wondering if you could tell us a little bit about your sort of overall research program and like what are you studying specifically in Stargardt disease?
Yeah, sure. So the key feature of Stargardt disease that causes the disease is accumulation of toxic molecules in the retina. And these molecules are derived from a compound called retinal. So retinal is a very, very important molecule. It's derived from vitamin A, and it's absolutely essential for the detection of light in the retina. So if you didn't have any retinal, you would not be able to see whatsoever. So retinal functions in the photoreceptor cells of the retina, and it acts very, very much like the spring on a mousetrap, okay? So when a mousetrap is activated, the spring closes very quickly, traps the mouse. So retinal functions exactly like that, except that light in the retina and photoreceptor cells will cause a retinal molecule to snap shut, just like a mousetrap spring. And this happens in about 1/1000th of a second, so super fast. And this change in shape causes signals in the retina to arise and signals to the brain to be sent, basically forming the basis of vision. Now, like a mousetrap, which has to be reset if you want to catch another mouse, the retinal molecule also has to be reset. But it doesn't just do that by itself. It actually undergoes a very, very complex pathway and process to reset itself. It actually has to be sent away from the photoreceptor cells to another cell in the retina called the retinal pigmented epithelium. Once it's there, then it can reset, and then once it's there, it has to go back to the photoreceptor cells. In Stargardt disease, that process is defective, and it leads to the accumulation of these retinal molecules, specifically in the retinal pigmented epithelium. And that leads to a toxic buildup of these molecules. And this buildup leads to its detection by the immune system, and specifically a very specialized branch of our immune system. So in our lab, we hypothesize that by blocking this part of the immune system, we will be able to block the inflammation that ensues from that and block the progression of all of the disease-related phenotypes and pathologies associated with Stargardt disease. So we're using mouse models of Stargardt disease, so a mouse that has mutations that are identical to those that are found in humans, and that they target the ABCA4 gene. And we're introducing into these mouse another mutation that blocks the formation of this protein that acts as the initiator of this immune pathway. And by seeing how these genes interact, we predict that losing this activator of the immune pathway will prevent the formation of Stargardts in these mouse models.
So in a simple way, would you say you're trying to reduce the immune reaction in the eye in response to?
In a nutshell, we're trying to completely block the initiation of that immune pathway and block inflammation.
So we heard earlier when Dr. Simpson was talking about gene therapy, about how the eye is maybe isn't as responsive to the immune system. So I was wondering about that, because we often talk about the eyes being immune privileged, which means we can do gene therapies and there won't be as much of an immune reaction. So where are these immune cells coming from?
Yeah, so there's actually a lot of evidence done from basic research, again, a lot of basic research done in using mouse models to tell us that a lot of these components of the immune pathway that I'm talking about are already expressed in the retina, in the photoreceptor cells, and in the retinal pigmented epithelium. So we believe they actually play a role in the homeostasis or maintenance of the environment and the health of the retina themselves.
So if you're trying to reduce the inflammation in the eye, would there be any other ways to reduce inflammation? Because I feel like it's maybe a trendy topic to talk about inflammation in the body or ways to reduce inflammation. Is there any evidence that any systemic approaches could also impact the inflammation? Yeah,
that's a really good question. I think we'll have to definitely examine systemic approaches like that. But I think in this particular case, it's going to be really well suited to kind of an approach, a targeted approach, because we believe that the effects that we're seeing are happening in very specific cells in the retina. So, for example, there was a trial a few years ago looking at geographic atrophy, looking at kind of similar types of inhibitors of inflammation. And, there's a good rationale for these experiments, but in those experiments, a lot of these inhibitors were targeted to the vitreal region, so a region that is not really maybe necessarily involved in the progression of the disease. So I think it's going to be important to really pay attention to where you're going to target. And as Dr. Simpson mentioned, the eye is a great system for trying these types of approaches because you can really target specific cells.
You mentioned geographic atrophy. So is there any maybe cross-pollination or work that is being done either in Stargardt disease that may impact treatments in geographic atrophy or AMD or vice versa?
Yeah, absolutely, because there are several features that are shared. I think AMD is a lot more complex than Stargardt to begin with. But things like autofluorescence of the retina and the formation of elevation of lipofuscin is found in age-related macular degeneration to some degree. So I think there could be some potential crosstalk there, definitely.
So sort of going back to the questions that we said we would ask you. Okay. I'm sorry about that. So you mentioned some of the research that you're doing and about the potential for a treatment. Is there any other treatments or therapies that are being studied now, either in development or even potentially clinical trials for Stargardt disease? Yeah,
absolutely. There's quite a bit of work done. People are trying gene therapy approaches for doing the approach that Dr. Simpson mentioned about augmentation therapy. But as was mentioned before, the gene for ABCA4 is very, very large, and it poses great challenges for that. Also, being able to target to enough cells is a challenge for that. There have been some work that's been done to actually see if you can modify vitamin A itself so that it can be generated in a form that can't form these harmful toxic molecules downstream. So that might be another interesting avenue to investigate.
Just thinking about that, so with vitamin A, obviously it's really important in vision. If you made a modified form, Could there be other impacts on the body, though? Or is vitamin A only used in the eye?
Exactly. So vitamin A is something that we need to ingest in our diet, and it's an important cofactor in all parts of our body. So that's the importance of pursuing basic research, is to be able to kind of probe those types of questions. So it's absolutely essential to do that in this case. Yeah.
And sort of thinking maybe looking 5 or 10 years in the future, maybe sooner, maybe longer, how do you see your research impacting patients with Stargardt disease? What are sort of the next steps in your research project?
Yeah, so the work that we're doing right now is pretty much a proof of principle type of an experiment. If we see a positive result with our work, that will really allow us to push forward with developing kind of therapies in the mouse that we can translate to humans, and that would be one goal. We're also in collaboration with Oak Bay Biotech, and this company has developed a small molecule inhibitor of the factor that we were working on, and we would like to push that forward as well through a clinical trial if we see positive results in our study.
So essentially doing the mouse study first, and if that is positive, looking then at a small molecule inhibitor.
Exactly.
Very exciting. Thank you, Dr. Chow. So we will come back to the whole panel at the end for more questions, but I'm going to move on to Dr. Orson Moritz, sitting at the end of the table to my left. So Dr. Moritz is an associate professor in the Department of Ophthalmology and Visual Sciences at the University of British Columbia. And Dr. Moritz's lab is trying to understand how gene mutations lead to damage in photoreceptor cells in diseases like retinitis pigmentosa, and hopefully using this information to lead to new therapies. So thank you, Dr. Moritz.
Thank you very much for inviting me. So I always love coming to these events.
We always love having you. So yeah, it's sort of a similar topics, helping to set the stage for some maybe individuals in the room who don't know about some of the specific inherited retinal diseases. What is retinitis pigmentosa, and what is the impact on individuals' vision?
Well, retinitis pigmentosa is a disorder of the eye that has a sort of specific set of symptoms that an ophthalmologist would report. And this involves a specific tendency to be very dependent on genetics. And it has a particular look when you look into the eye, that you'll see some pigment deposits in the eye. And the patients tend to get a tunneling of their vision. They lose their peripheral vision first, and progresses to a loss of their central vision. And so this is a sort of unifying set of symptoms, but the underlying genetics is very complicated. There's many, many different genes. Unlike Stargardt's disease, where it's mostly caused by one gene in particular, retinitis pigmentosa, there's many different genetic causes. So you have dominant retinitis pigmentosa, you have recessive retinitis pigmentosa. Each of those is caused by a number of different genes and many different mutations in those genes.
So that sounds like it could be very complicated, both in terms of diagnosis, but also in terms of potentially finding treatments if you have so many different genes and gene mutations.
Correct. So that's actually kind of one of the focuses of my lab, is we're trying to understand if you have all these different genetic causes, but they all come out looking like something that's kind of the same. What do they have in common, and what separates them? So maybe thousands of different genetic mutations cause retinitis pigmentosa, but does that mean there's 1,000 different diseases, or is it one disease, or is it 3 diseases? Or the smaller the number is, the better answer, I think, right? Do you have to have 1,000 therapies, or do you have to have one therapy or something in between?
So I was wondering if you could give us sort of like set the stage about the sort of therapies that are there any therapies available currently for retinitis pigmentosa?
Well, there's a few controversial ones. So there's vitamin A therapy, but there's sort of limited benefit has been shown for vitamin A therapy. And in addition, taking large doses of vitamin A is actually not that good for you. So there's a bit of a controversy as to whether the potential benefit is worth the health risks of taking that much vitamin A. And there's a few things that have been tried for very advanced retinal disorders, like these retinal prosthetics. And they're, I think, also shown to be a very limited use, and in some cases they've been taken off the market now, I think. So really the number of therapies is close to zero, if not zero.
And maybe that is a good lead into your research. So what is your research around retinitis pigmentosa, and sort of how are you hoping that could potentially impact on therapies?
So like I said, I think one of the things we have this program where we model retinitis pigmentosa by making genetically modified animals. And unlike a lot of people who use mice, we use something that's much easier to genetically manipulate. It's a frog. And yeah, it often generates chuckles as well. And it is very, very easy to genetically manipulate these animals. And so what we can do is, at this point in my lab, we've probably made upwards of 50 different models of retinitis pigmentosa. So most people would spend their lives studying, one or two types of mice, but we generate lots of these animals, and then we ask the questions, how are they similar and how are they different, right? Within that system, we've been able to generate descriptions of several different classes of mutations in rhodopsin that cause retinitis pigmentosa. The rhodopsin gene is one of the major causes of dominant retinitis pigmentosa. So we've been able to identify at least 3 and probably 4 or 5 now. In recent months, we've seen some signs that something else is happening of these mutations. So there are about 100 different mutations in rhodopsin that cause retinitis pigmentosa, but we can say, well, they all belong to, sort of cause 1, 2, 3, or 4, not 100 different causes. The other thing that we've been looking at that's we find very interesting is that in a certain subset of people with retinitis pigmentosa, their retina is not symmetrically affected. That's very interesting to us. So if you have the lower part of your retina more affected than the upper part of your retina, but all of the retinitis pigmentosa in that retina is caused by one mutation, what happened to the upper half of the retina that protected it? Or what happened to the lower half of the retina that made the problem more severe? If you know that, it suggests there's already the possibility of preventing that particular type of retinitis pigmentosa. So what we've been looking at is sort of environmental and dietary influences like vitamin A on those particular subsets of retinitis pigmentosa.
So a few actually questions about the frog research, because I haven't talked to too many researchers who do work in frogs. So is the frog eye very similar to the mammalian or to the human eye?
Well, in some ways it's very similar. It has the same basic types of cells. It has rods and cones and retinal pigment epithelium, and it has axons that go to the brain by an optic nerve. So it has all those basic features of a vertebrate eye. It has some major differences as well. So the photoreceptor cells are much larger, which actually makes them very easy for us to study using microscopes. And the retina develops very fast. So if we don't actually study really the frogs, we study the tadpole offspring of the frogs. We breed the frogs. They'll lay 1,000 eggs, and about 3 days later, the little fertilized eggs start to develop eyes. And then about 2 weeks after that, the eyes are completely mature. They're only about 1 millimeter across, but they're completely mature. So we can do an experiment, in a matter of a week or so. And that kind of accelerates things a lot. So, on the other hand, there are, problems with using a frog. You're always wondering, is my frog reporting on what is actually happening in a human, right? So we have to sort of reassure ourselves about things like that. Like, for example, when we put mutations that cause retinitis pigmentosa into a frog, they cause something that looks similar in the frogs. So in that way, we sort of have some confidence that what we're doing is valuable.
I also thought it was really interesting when you were talking about how the potential impact of environment or other maybe gene interactions, about why parts of the retina might be more affected than the other parts. So do you have any other indications, environmental or genetic, about what could be causing this variation?
Well, yeah, so I'll tell you, one of the experiments that we've done that's, I think, just so interesting in the result is that we suspect that one of these environmental influences is light. And in a lot of scenarios, we can change, in these animals that have these same mutations that these people have, we can change the rate at which the retinal degeneration happens by changing the lighting environment. So we have one class of mutations where if we keep the animals in the dark, the degeneration slows down a lot. We have another class of mutations where if we keep the animals in the light and never turn the lights off, it slows the degeneration a lot. So you can see the end result in the human looks very similar. But the actual, if you sort of dissect out the underlying causes, they can be very different.
I think that's really fascinating because I know protecting from light is really, really important for many people who have inherited retinal diseases. And it's interesting that there might be a subset where, not that it's not important, but that it's maybe more complicated than we think. It's
complicated enough that I don't think I can give specific recommendations at this point. Do you know, if you know what I mean? I know there's something there, but it's premature to say, I suggest you do this. If you know
but hopefully soon.
Yeah.
Hopefully soon. And finally, I was wondering if you could just tell us how you came to study retinitis pigmentosa. What brought you to this field?
So actually, I was undergraduate student. I was working with Dr. Bob Molday, who's not here today, but he's often also at these events. And I joined his lab as an undergraduate student, and it all just seemed so fascinating to me. And the thing that we were studying, the retina, was so sort of elegant a research subject, and the things that you're studying are so impactful, blindness, and these things just sort of go together as being a really fascinating thing to kind of spend your life on, I think.
Thank you. So thank you, Dr. Morris. And again, of course, going to continue sitting there, so there will be time for questions at the end of the panel. And finally, we're going to turn to Dr. Kevin Gregory-Evans. So Dr. Gregory-Evans is an ophthalmologist and macular researcher. He's a professor of ophthalmology at the University of British Columbia as well, and the Faculty of Medicine. So welcome, Dr. Gregory-Evans.
Hi.
Do you mind just moving a tiny bit closer to the microphone? Thank you. So maybe a little bit differently than Dr. Moritz and Dr. Chow, you are a clinician scientist, and your study is potentially broader. It's not as focused on one or two different diseases. So I was wondering if you could explain a little bit about the type of research that you do and how it links to your clinical practice.
So the type of research I do has evolved a lot over time because, as you say, being a clinician scientist is a pretty broad remit, I think. We often get accused of being a jack of all trade, a master of none sort of approach to things. But usually people then narrow into a particular area that they're interested in. When I started my research career, I was very much into gene discovery and identifying families. The first project I did was to identify a disease gene in a very large family with a cone rod type retinal dystrophy in Merseyside in Liverpool in northern England. Since that time, though, I then moved into studies of the molecular biology of retinal degeneration, then into stem cell biology, and now I'm mainly focused on clinical trials. Clinical trials currently for gene therapy. to some extent pharmaceuticals, but mainly gene therapy trials. So that's where I am at the moment.
And a great lead into the question about what is a clinical trial. I think we all have an idea of what it is, but I was wondering if you could give us a little bit more detail about the types of clinical trials there are.
Yeah, I think that this is quite an important question because to some extent, the concept of the clinical trial has been hijacked by the pharmaceutical industry. So if I was to ask people in the audience what they thought a clinical trial was, most of you would think that it's a big thing, that it involves lots of people, it is really expensive, and is all about finding a treatment. And if that clinical trial doesn't work, that means that that treatment doesn't work. That isn't the case. That's just one type of clinical trial. So even for things like retinitis pigmentosa and macular degeneration, if you look at the history of clinical research, most of the real benefits, most of the things that have led to real innovation in ophthalmology have not been these large, multi-phase, pharmaceutical industry-dominated clinical trials. So, for instance, there are probably a third of the audience here have had cataract surgery. So the initial studies in cataract surgery didn't involve a pharmaceutical company. It wasn't even a big study. It didn't even involve ethics approval by an institution. It was just a small study done by one ophthalmologist in London, at St Thomas's in London. Another treatment, Avastin for wet macular degeneration. A lot of people with macular degeneration in Vancouver are treated with Avastin. Avastin didn't come about through a large prospective double-blind trial sponsored by a pharmaceutical industry. If anything, the pharmaceutical industry didn't want Avastin to be used in macular degeneration. And it was an ophthalmologist working on his own in Florida that started the trials for Avastin. So it is important to remember that clinical trials have a much broader remit. There's case series where one person just documents what they're finding in a small group of people. Then there are these small clinical trials that are university-led by researchers like us. They can produce, in some ways, a lot more positive information than is possible with a large-scale pharmaceutical study. So if you go from a basic science study immediately into one of these large pharmaceutical company trials, I think a lot can be missed by doing that. It's a bit like, I was trying to think of a good analogy this morning about what that jump in innovation is like. So for instance, penicillin was discovered at my university, Imperial College in West London in the 1930s. If penicillin had gone from basic science by Alexander Fleming straight into a big clinical trial, that trial would have failed because there were lots of innovations that were needed in addition to the basic science to get penicillin to actually work in the 1940s. The two particular problems that they had with penicillin right at that beginning stage was that you need large volumes of penicillin at that time, the way it was being made, large volumes. It wasn't a tablet at that stage, it was more like food, like eating a chocolate bar every day full of penicillin. And the other problem was that the body exuded it really quickly. So if you'd had a big study at that stage before those problems had been solved, it wouldn't have worked. That trial would have failed. And I think that's true of some of the clinical trials that we're seeing now, that the expectations are far too high. I was involved in a gene therapy study, I don't know if it's already been mentioned, in choroideremia. And that study technically failed. But that was because the expectations of that study, in my opinion, were far too high. So I think it's important to remember that there are other types of clinical trial. When you do a large study, a pharma-sponsored study, there are also these phases that are asked about. So a phase 1 study is mainly about checking whether the drug is, or the gene therapy is safe in humans. And despite the large emphasis on mice and tadpoles, a human is not a tadpole. And that is particularly true when it comes to side effects of pharmaceutical agents. You can see things very commonly in humans and not see them at all in animals. So that phase 1 study is quite important. The phase 2 study, again, is about safety, but this is when the sponsor will start to gather statistically significant information on efficacy. So efficacy is really, really important, and safety and efficacy are the two main problems in getting from a starting clinical trial into FDA approval, Health Canada approval, and into clinical practice. So when you think about efficacy, you can think about it this way. If I had a treatment that would make you taller, how many people would like that treatment? It was guaranteed to work, and it was guaranteed to have no side effects. Yeah, I think a lot of people would ask for that, would want it, and I'd be really rich. But what if I said to you that it cost $1 million, like gene therapy is going to cost? There are people that would then say, no, I don't want to be taller that much. But I'd still have a lot of patients. A lot of people would want that, like a lot of people want to have gene therapy. But what if I said to you that the guarantee in increased height was only 0.5 centimeter? And that's the problem that clinical trials have to deal with, that a lot of things that work very well, seem to work very well in animal models, when it comes to humans, the effect is so small as to be not worthwhile. So that's why you have this phase 1 and phase 2 is so important. Phase 3 studies, and these are the sort of studies that I would encourage people to be particularly interested in if they want to enroll themselves in studies. Phase 3 studies, the pharmaceutical company is fairly confident that they have something that works and fairly confident that they have something that's safe. And essentially, the phase 3 study is to, on as large a population of patients as you can get, in order to find these rare associations, these rare complications. And then we have phase 4 studies, which are after FDA approval. This is a drug now that is accepted as working, is accepted as safe. And this is sort of ongoing monitoring for the first few years of a drug's use. You could say that Luxturna, to some extent, is at that stage right now and will be monitored probably for the next 5 to 10 years to see whether the benefits that you get with Luxturna carry on beyond that.
Thank you. That was a great overview of the different types of trials. So a last question before we move to audience questions. You mentioned a trial that you were involved in with choroideremia clinical trial. Do you have any other clinical trials, either that you're involved in or that are taking place in Canada, that you think the audience might be interested in hearing about, not necessarily joining?
I think it's already been mentioned that there are a lot of clinical trials around the world focusing on gene therapy for inherited eye diseases. There are a few clinical trials that are targeting other approaches, like stem cell use, electronic retina, optogenetics. But I think that currently, the ones that you should be paying most attention to are the ones that are focused on gene therapy. They're the ones that are going to be the most benefit in a time frame of the next 5 to 10 years. The trials that I'm involved in are targeting choroideremia, X-linked retinitis pigmentosa, dominant retinitis pigmentosa, and we are about to start one on X-linked retinoschisis. So there are studies in Canada. It's important to remember, though, that when Donald Trump was in office, one of the things he said was that America, or the United States, is the research laboratory of the world, and we should be grateful for that, and perhaps contribute to it. And to a large extent, he was right when he said that, particularly when it comes to clinical trials. Ones that are going to be truly beneficial to people often start in the US, because their research, especially their commercial research organization, works in a very efficient industrial way that other countries in the world just can't put together. So most clinical trials start there. There's potential for Canadians to be involved in those trials, but one of the saving graces that we have when it comes to clinical trials in Canada is that when a pharmaceutical company has something that seems to be working or is very rare, like a lot of inherited eye diseases are, the American companies always have to go outside of the US to recruit, and they often come to Canada first. So we tend to be involved in studies that are phase 2 or phase 3. So those studies where there's just nothing here worthwhile don't usually come to Canada, which is a great benefit to us, I think. So there are trials coming, and there will be lots.
Thank you, and thank you to all of our panelists. And we have a few minutes before lunchtime, so I'd love to open it up to the audience if the audience has any questions. Morgan is coming around.
Hi there, thank you for coming today. One question I wanted to kind of find out about is, I'm living with retinal pigmentosis. Rhodopsin is the gene of my issue. I was just wondering, there's a lot of research on acupuncture when I read online, because I'm trying to delay the progression of what I have. As you said, that tunneling vision just keeps happening over and over and over again. Do you have any insight into acupuncture, and has there been any studies on it?
I've seen it mentioned, and I think there are some people in Vancouver who specifically do this therapy. I haven't seen any evidence that it works. And I know there's, actually I did review, like Elizabeth, review the clinical trials, ongoing clinical trials, 200 pages of, clinical trials or 200 entries for clinical trials on retinitis pigmentosa. And there's at least one group doing a clinical trial on acupuncture, I think again in the US somewhere. So I haven't seen any evidence that it does work. Just off the top of my head, as a person researching retinitis pigmentosa, there's no obvious reason to me why it would work. But it's got enough interest that somebody is doing a sort of comprehensive study on it.
Got it. The other follow-up I have is, for these clinical trials, is there a way for us to just sign up ourselves to be involved in any of these trials, or does it always have to go through the doctor?
Probably Kevin would be the better one to answer that.
I'll just add some comments about acupuncture and retinitis pigmentosa. So acupuncture is not a scientific treatment in the Western sense of science. It has never gone through any form of rigorous Western scientific interrogation, and it probably won't. I've got a number of patients that are having acupuncture often combined with traditional Chinese medicine. So acupuncture is an example of a priori type research in that people have an idea that something can work. They can see a mechanism by which it could work. Therefore, why bother testing it? It's a treatment. It must work because I've worked out how it works. That's a very Eastern approach and a very Russian approach to science. Another example is stem cell therapies. You can go to a stem cell therapy clinic with retinitis pigmentosa right now and get that therapy. It'll cost you a lot of money, but you can get it. But there is no proof that that technology works yet. But the idea of stem cells is very persuasive. So it's a very different, acupuncture is done in a very different way. The ideas behind it, supporting it, are not Western scientific. Now, I would add to, just to add to that, that doesn't mean it doesn't work. It means that by the standards of Western medicine, we don't know it works. And we can't say that it does. That doesn't mean it doesn't, but we can't say that it does. So when I get patients asking me about it, I say, well, if this is something that you want to do, then do it. But I'm not recommending it. And I have patients that have said very good things about the therapy in Vancouver, and others that have said the complete reverse. When it comes to getting involved in clinical trials, there is an avenue for you to go as an individual patient to enroll into studies. And that's through a website called clinicaltrials.gov. There they will list clinical trials and will give you contact information. The good thing about clinicaltrials.gov is that the studies that they list are legitimate scientific studies and are looking to answer questions in a Western scientific way. So they're bona fide, proper studies. The downside of it is clinicaltrials.gov isn't really designed to recruit patients. It's more about recruiting sites to be involved in clinical trials. And that's why usually it is possible as a patient to be recruited, but it's usually better to be recruited through a clinical site, an ophthalmologist or a researcher. Then it tends to be easier for you as a patient to get involved.
Thank you.
So Dr. Chow, you mentioned before with Stargardt's that you've got some research that shows that smoking and blue light will impact Stargardt's. And I'm just wondering, if there was research to show that, and would that be similar for RP as well? Because I know they're very different diseases and different genes, and I just wondered if those kind of environmental factors have been shown to prove impactful on RP and the degeneration of eyesight.
Yeah, I think the mechanisms will be different. Stargardt disease is specifically related to the accumulation of lipofuscin, which is this vitamin A-derived molecule. And that molecule itself, we know is highly reactive, lipofuscin. With blue light, it will be converted to reactive oxygen species. And those are actually the molecules that cause the damage to the cell. And that kind of mechanism isn't really happening in RP, so it'll be separate. Yeah.
Thank you very much for this informative panel. Just two questions regarding RP. One, could you say something about the prospects, for example, for electromagnetic stimulation? It's advertised in the U.S. as one of the venues. And the second one is optogenetics. Are these potential cures for RP?
Yeah, I think the optogenetics is very interesting thing that hasn't been brought up yet this meeting, at least not since I came in this morning. So that's a very interesting type of gene therapy that is directed at treating a retina which has lost its photosensitive cells. And so one of the issues that you have with these gene therapies is that in a lot of cases they'd be directed at a young retina that hasn't developed the disease yet. Once you've developed retinitis pigmentosa, a gene therapy of the type where you just replace the missing gene isn't going to reverse your progress. At best, it's going to delay further progression of the disease. But optogenetics sort of holds this promise of we'll make the other cells of the retina that aren't normally photosensitive, we'll make those photosensitive instead. So it's introducing a new functionality by a gene therapy to the retina. And there's quite a lot of interest in that. And I think there are also clinical trials of that therapy that are being, if not initiated, that they're sort of in the planning stages. And a lot of people are quite interested in that. The electromagnetic stimulation, honestly, I'm not as familiar with that, and I don't have a lot of information on that. Are you talking about a prosthetic retina, or are you talking about just electromagnetic stimulation of the eye with a device?
Yeah, that's what he means.
Yeah, I don't really know much about that.
I can tell you
honestly.
a bit about that. So electric stimulation is based on the concept that if you traumatize the eye or traumatize the retina, it'll release neurotrophic factors that will help to protect the retina from further damage. And it's a direct, you electrocute the eye.
Yeah, yeah.
There's a lot of basic science to show that there is this effect. There have been some human trials. Those trials have been positive, mainly from the point of view of safety. That's what's been reported, that it's a safe procedure. You wear a sort of form of modified spectacles to electrocute yourself, sort of thing, with a very low current. I think that the state of play at the moment is that there is a therapeutic benefit, but it doesn't seem to be very large. So if they can increase the therapeutic benefit, there might be something in it in the future, but it's still at the stage of seeing if there's enough. It's like a phase 1, phase 2 study. They're not certain yet whether there's enough therapeutic benefit to make it beneficial. The thing about optogenetics is that there's a difference between curing a condition and treating it, okay? Most therapeutics that we have across medicine are not cures, they're treatments. So, for instance, currently for diabetes, nobody's been cured from diabetes, they're being treated, okay? There's a big, big difference, that there are things that you can give that are therapeutic benefits, okay? Optogenetics will never be a cure for vision loss. They can be a treatment to some extent, but the basic concept isn't there for it to be a cure, because you're modifying the retina in a very complicated way. You're trying to use the retina in a way it was never designed to be. So when you genetically modify these other cell types, there's a reason that the retina has many more photoreceptors than any other cell type. There's a reason for that. And that has a direct effect on the quality of vision that you're going to get. And that's why optogenetics is an interesting treatment, but it isn't a cure. Not a cure in the way that perhaps a gene therapy can be a cure one day, or stem cell therapy can be a cure one day.
Okay, we have time for one more question. We'll take back here.
Hi, thank you so much for all the information that's been shared today. I really appreciate everybody's skills and knowledge and passion. My question is a broader question, and I guess it goes to the process. And what I'm wondering, and what's missing for me is, there's lists that have been shared of all the numbers of gene therapies that are in trial or at some phase. What I'm trying to understand is how did that list come to be? Like, is there a governing body that chooses a project, or is it based on curiosity? I guess that's the part of the process where I guess I'm missing and not quite understanding.
This question came up in the past at a panel here, and somebody else on the panel disagrees with my opinion on this. I would argue that I would argue that there's no oversight over what studies are done, where they're done, who does them. There is no overriding governing body. There's nobody out there that says, well, the commonest cause of retinitis pigmentosa is X, therefore we're going to prioritize that. It doesn't exist. There are influences at play. Because at the heart of research, there are two important factors, one more important than the other. One is money, and funding bodies can direct research projects. That in itself is thought of as quite a controversial thing, because scientists like to do open sky research and be allowed to do whatever they like. But there are funding bodies that do direct research in particular directions, which I think is a very, very good thing. The second thing then goes to the passion of the researcher. And as Orson describes, he got interested in getting involved in retinitis pigmentosa because he was inspired by Bob Moldey and the people around Bob Moldey. And that's an important influence as well that can direct research down a particular path. But when it comes to oversight that perhaps you'd think of in a government way, there is no oversight.
I mean, I think another thing that I would say is there are certain problems that appear on the outside easier to solve. And I think that's the reason why you have a gene therapy, Luxturna, for an extremely rare eye disease right now. It's because that particular eye disease was kind of a sitting duck in terms of a gene therapy target. It has several unusual properties. It's a missing gene. The gene is defective, so all you have to do, you have to put the gene back. It has an unusual property that the gene is missing, but the retina doesn't die off rapidly. So you have adults that have still a lot of retina present. If you put the gene back in, there's a lot of retina that could function. And it had a dog. There was a breed of dog that coincidentally had this exact same problem. And so people could test this gene therapy on dogs very easily. And then there was a good prospect that if you treated an adult, that you would have an effect. And it's, you know, for ethical reasons, it's much easier to do studies on adults who can consent to the idea that I'm going to have viruses injected into my eye and with potentially disastrous consequences, although hopefully not, as Elizabeth Simpson told us about, right? So this particular disease, although it wasn't, you know, a very widespread genetic disorder of the eye, became the first target because of it was a target that people could work on very effectively. And that often happens, that, you know, certain advances suddenly open up the possibilities that, oh, now, you know, we've seen this, now we can do this. And it's not necessarily a route that follows things like exactly the most common eye disease will be the first one to get a treatment. Probably not.
Okay. Yeah.
Something from my perspective, or? No, go ahead. Just also, your question about how does this all happen and how to get these different types of programs set up. Sometimes we don't set out at the outset just to treat a specific kind of disease. There are clinicians that are going to be focused on patients with certain diseases, and there will be basic scientists. And 10, 20 years ago, there was a great divide between the two. Basic scientists, such as myself, are very much interested in looking at understanding mechanism and just trying to understand what is happening in the system. Maybe not necessarily with a disease, a specific disease in mind, but just understanding the system. And as we learn from those types of research, we actually can converge with clinicians and find commonalities and then use those to specifically identify diseases from basic research and pursue those. So sometimes things arise just due to attaining knowledge.
Thank you. I think that was actually a really nice ending to this session about sort of the power of research and the complexity of research. So thank you, Dr. Moritz, Dr. Chow, Dr. Gregory-Evans.